Very close to the last keynote of the conference, so I wanted to make a couple of announcements before we finish. So first of all, a lot of you have been getting emails about the top 10 reading list of papers in regulatory and systems genomics. So the goal for this is an activity that we've been doing for the last few years with Saurabh Sinha and myself. This is one of the activities that we thought would be really beneficial to the community. So the goal is to build a reading list of papers that represent our field that are really recent. So we want to pay attention to landmark papers as they come online. So we don't want to simply sort of discover them 2 or 3 years down the road, but see them come over. Introduce outsiders to papers that are relevant to our community, as well as highlight papers of interest to other members of our community that may have simply not noticed these papers. So we solicited community suggestions from past attendees of this conference. We received votes from the community. We also, so I have to admit that last year we simply went based on the votes. This year, the votes were extremely close. Everything was basically voted just very, very highly. So we needed additional metrics to make it more meaningful. So I have to admit that we actually did look at citations which were normalized for the date of publication. We looked at the journal impact factor for our field, namely what are the most relevant journals to our field. We looked at the relevance of these papers to our conference and to our community. And we also accepted personal bribes in the form of spiced chocolate. So just so that you know, future years. And based on that, we basically came up with the following list. So these are the top 10 papers. So this is a paper by Greenleaf that introduced the attack-seq method, so transposition of native chromatin for fast and sensitive epigenome profiling. This is a single molecule dynamic of enhanced enzymes by the Liu Lab, published in Cell, characterizing the genetic base of transcriptome diversity by Battle et al. From the Color group in Genome Research. Comparison of D. melanogaster and C. elegans by Lee et al. That I think you just heard from the Brenner group in Genome Research. By the Hughes Lab, determination and inference of eukaryotic transcription factor sequence specificity. A community effort to assess and improve drug sensitivity prediction. So this is one of the DREAM papers in Nature Biotech. Co-regulation of transcription factor binding and nucleosome occupancy through DNA features of mammalian enhancers by Barozzi et al. From the Natoli group in Molecular Cell. Another Nature paper from Furlong group looking at enhancer looping in development and polymerase association. A paper by, again, a member of the conference here, so TFBS Shape, motif database for DNA shape features in nucleic acids research, as well as a paper by Julien Gagneur in the Steinmetz group in PLOS Genetics. Again, given how close the voting was, I had to put the runner-ups here, apologize. So again, you can see the runner-ups are really, truly outstanding as well. So by the Regev group, Kramer in Cell, and so on and so forth. So I won't read all these. We'll link them on our website. We have Google Scholar links for each of them that you can just use. Again, this is a reading list. I encourage you to just peruse these papers and keep submitting them for next year. The last announcement I want to make is that RECOMB, Regulatory and Systems Genomics with DREAM, I think we're at 10 now, in 2015, will be in Philadelphia between Monday to Friday, November 16 to 20. So mark your calendars. This is a tentative date. I don't want to tell you, hey, this is it, but this is pretty close to it. So we're still, figuring out the details, signing the contract, and so on and so forth. So Philly is an absolutely beautiful town. So you'll possibly recognize the Liberty Bell. And really, truly historic city, absolutely gorgeous landmarks, and a real pleasure to be there. So it's the week right before Thanksgiving, so we didn't want to interfere with the holidays. We're also going to possibly try to squeeze the conference into 4 days, 1 day for DREAM and 1.5 for both regulatory genomics and systems biology each. And we hope to see you all there. Before closing, I want to thank Diane Kovacs, who's sitting right there in the back. On behalf of all of us, thank you. So this meeting has been a huge, huge amount of work, and the reason why it's even possible with 3 organizers who are as busy as we are is that Diane and Chris, I want to really personally thank Chris. And of course, our session chairs, Christina, who's right there. Thank you, Christina. I think Lonnie had to run off, but let's thank Lonnie Welch. And of course, Gustavo and Andrea, who have just been spectacular year after year. So please thank them too. All right, that's all I have to say. Now, without further ado, I wanted to